In this class, let us talk about intragenic interactions. I mentioned already what do you mean by that one. It is nothing but the interactions that occur between the alleles of a same gene, that is between capital A and small a, like that. It is also called intralocus interactions or intra-allelic interactions or simply allelic interactions. The examples for intragenic interactions as given in the book. Number 1, incomplete dominance. Incomplete dominance. This is one example. Incomplete dominance. Number 2, codominance. Codominance. This is number 2. Number 3, lethal genes. Lethal genes. Number 4, polygenic inheritance. Polygenic inheritance. And number 5, pleiotropy. Pleiotropy. So in the case of incomplete dominance, codominance and lethal genes, interactions occur between a pair of genes. In polygenic inheritance, a single character is controlled by many genes. In the case of pleiotropy, it is called one gene, many characters. Here polygenic inheritance, many genes control one character. In the case of pleiotropy, a single gene controls the expression of many characters. We'll talk about later in detail. Now we'll take the incomplete dominance, how far the character occur. Now in the case of incomplete dominance, there are two alleles and one allele is not completely dominant over another allele. There are two alleles. One allele is not completely dominant over another allele. And this is called incomplete dominance, the phenomenon. So let's take the example and then we'll discuss. We have the following examples for incomplete dominance. 1. Color in 4 o'clock plant. Flower color in 4 o'clock plant. That is the binomial for this plant, Mirabilis jalapa. This is one example. Another example, again the flower color in the case of Antirrhinum. Another plant, Antirrhinum. Antirrhinum majus. Here also the flower color. This is commonly called as a snapdragon or dog flower, because in appearance it is looking like the sitting posture of a dog. So these are the two examples related to the flower color, that is in plant. There is also another example in the case of animals. One fowl variety called Andalusian fawn. This is one bird variety, Andalusian fawn. There the feather color. Here the feather color is taken into consideration. Feather color. So these are all the examples for incomplete dominance. Flower color in Mirabilis jalapa and also Antirrhinum majus, and the feather color in the case of Andalusian fawn, a type of fawn variety. Now let's go for the explanation with an illustration. The process of incomplete dominance can be explained with the help of flower color in Mirabilis jalapa, commonly known as a 4 o'clock plant. Here there are two varieties of flower color. One is red, another one is white. Now, as neither of them are equally dominant, we are taking actually the same genes capital R with R1 and R2. We are not taking the recessive letter. So when cross is effected between red flower plant with white flower plant, the F1 generation, the character actually did not exhibit the parental variety. No parental variety is being produced in the F1 generation. Okay, the character did not exhibit, actually this cross we can see, this cross did not exhibit the dominant trait and shows what is called incomplete dominance. In incomplete dominance, one allele is not completely over another allele. Now let's take a cross, the gametes formation. In the F1 generation, we have formed a hybrid with the genotype R1, R2. It is heterozygous. But the nature of the color of the flower is pink. The reason for that one, the R1 allele, capital R1 allele, is not completely dominant over R2 allele. That is the result. We have the expression of, that is a new character, an intermediate character between both the pure varieties of the plant. See here, when F1 hybrids produced the gametes, so we have two types of gametes on either side. So we are getting the probability of 4 individuals with the genotype 3:1 in the case of monohybrid class, but here the phenotypic ratio is 1:2:1, and so also the genotypic ratio. That means in complete dominance, that is between tall and dwarf plant, that is height of the plant, we are getting the ratio 3:1. But in the case of incomplete dominance, as one of the allele is not completely dominant over another allele, here capital R1 just over R2, we have received normally 3 different phenotypes, red, then we have the 2 parallel types red and then white along with pink. So red, pink and then white in the ratio of 1:2:1. We are getting the phenotypic ratio 1:2:1, and so also this is the genotypic ratio 1:2:1, and the phenotypic ratio also 1:2:1. So we have received the appearance of both the phenotypic and genotypic ratio same in the case of incomplete dominance. Now we have to go for further explanation. So for that I have to represent what we have the F2 generation. What are the gametes produced and what are the individuals produced? What is the genotypic ratio, phenotypic ratio that will be represented in Punnett square. Heterozygous individuals are pink colored. When they produced gametes, we have 2 different types of gametes, 50% with R1 and 50% with R2. By means of segregation and random combination, we have received 4 individuals. Out of the 4 individuals produced in F2, one is with red flower, another one is with pink flower, another one is with white. While we are looking into the phenotypic ratio, out of 4, one is red and 2 individuals are pink in nature and one is white. A genotype with R1 produced red color, a genotype R1 R2 results in pink color, and the genotype with R2 produced white color. So in the case of such processes, we have received the phenotype 1:2:1, and so also the genotypic ratio 1:2:1. So if in a hybrid class the phenotypic and the genotypic ratios obtained is same, that is 1:2:1, we can say it is because of incomplete dominance. Now what is the reason? Now the R1 gene, this is responsible for the production of normal enzyme for the synthesis of actually the flower color. And R2, it produces a defective enzyme. R2, a defective enzyme. So it is unable to synthesize what is called the flower color pigment that becomes white. Suppose what are the conclusions from this class? The following conclusions were made from this class. Number 1, the 2 alleles in a heterozygous condition, the 2 alleles themselves remain as discrete and unaltered. Then proving the law of segregation. Proving the law of segregation. This is one conclusion. Because of the segregation only, the two factors get segregated. Once again, the parallel characters reappeared in the F2 generation. So the two alleles themselves remain as discrete and unaltered units, proving the law of segregation of matter. Then, the second one, there is no blending of genes. No blending. No blending of genes. This is the second one. The third one, if you are taking actually, so no blending of genes. The phenotypic ratio is equal to genotypic ratio. These are the conclusions made from this class. Number one, that is the two alleles themselves remain as discrete and unaltered units. Actually, there is no mixing, there is no blending, proving the law of segregation. So I mentioned already during the gametic formation, the F1 individuals segregate and also recombine to produce the individuals in the ratio of 1:2:1 as red, pink and white. This is because of law of segregation. This is the conclusion. And there is no blending of genes. Suppose if blending had taken place, imagine that a blending had taken place. So the two pure varieties would not have appeared in the F2 generation. If blending had taken place, the two pure breeding plants, the parent plants, would not have appeared. And all the F2 plants would have only pink color. So this is because of that conclusion. We can just say that there is no blending. If blending had taken place and the pure breeding plants would have not appeared in the F2 generation, and all the F2 plants would have just actually pink color. This is another conclusion. So normally this cross actually give a conclusion that it follows the particulate inheritance of Mendel. The particulate inheritance of Mendel occurs in this class. And also it is confirmed the reappearance of what we call the pure breeding parallel combinations. So this class normally just giving a conclusion that a particulate inheritance takes place, and because of the particulate inheritance only, we have formed the two parallel types once again in the F2 generation. This is being confirmed. So that's about what we call the incomplete dominance. How far actually, what is the importance and what is the significance? Two alleles actually remain together without mixing, no blending. Then the phenotypic ratio is equal to genotypic ratio, and it would not have appeared what we call the pure breeding varieties. Then we can say that is blending occurs, but here blending had not taken place. If blending occurs, you know that one, we have only pink colored form in the F2. But the appearance of these three varieties indicated that no blending of the genes occurred. So we have formed the three different varieties, the reappearance, the F2 generation, and also the particulate inheritance of Mendel took place in this class, and that is confirmed because of the reappearance of both characters in the F2 generation. Now the molecular explanation for incomplete dominance can be explained based on the gene expression in a quantitative way. That is, how much amount of the pigment produced, either the red. Accordingly only we have the red color flower or the pink color flower or white color flower. This is what's called quantitative way. Amount of pigments produced. Now we have a wild allele. The wild allele is the dominant allele. The normal allele is called wild allele. This is R1, which encodes for a functional protein. functional protein, which encodes for a functional protein. So this encodes, coding encodes for functional protein, nothing but the enzyme. So it is a functional, encodes for functional protein. So it is a wild allele but functional allele, and which helps in the synthesis of the red pigment. So helps in the synthesis of red pigment. This is number one. This is a normal allele. Now another allele, what we have, that is the mutant allele. The mutant allele. The one which results because of a mutant or mutation, because of mutation, that is R2. So it codes for enzyme, codes for enzyme, encodes for enzyme, but cannot synthesize, red pigments, So it is caused, actually the mutant allele is produced because of mutation. So that we have received the mutant allele R2. Though it codes for and encodes for enzyme, but cannot able to synthesize what's called the red pigment. Now if you are taking actually red color flower, it is because of the genotype R1. So red color is produced because of the genotype R1. And white flower is produced because of the genotype R2. And if you are taking the pink color, that is the intermediate color, it is produced because of the heterozygous condition of R1. Now here 100% pigment, red pigment is produced. Here there is no pigment is produced. But if you are taking actually intermediate pink color variety, we have two alleles. One allele is R1. It is a functional allele. But out of the two alleles, only one functional. So this allele produces only 50% of the functional protein, that is enzyme. So in heterozygotes, the R1 allele produces only 50% of the functional proteins, that is the enzyme, resulting in half of the pigments of the red colored variety. So we have the pink color. So only 50% expression, resulting in 50% functional protein. So half of the pigments of the red color is produced, leading to what is called the pink variety, only 50%. So if we have this intermediate variety, that is the intermediate pink phenotype, with 50% functional protein, cannot create the red pigment phenotype homozygous. Red pigment phenotype homozygous. Because the red pigment homozygous variety has 100% functional protein. So that we are getting what is called red color. I'll repeat once again. Here there are two copies of R1. So that we are getting 100% synthesis of the red pigment. First 100% synthesis, 100% actually the production of the protein, the enzyme, which results in 100% production of the red pigment. The second case, enzyme will be produced, but there is no production of red pigment. That is why the flowers are white color. In the case of intermediate variety with pink color, only we have R1 alleles, functional. The R1 allele produces an enzyme, that is 50% functional protein or enzyme, resulting in half of the pigment formation of the red plant. So it becomes pink. So in the case of intermediate variety with 50% protein, homozygous actually the phenotype, 100% that is red pigment cannot be produced, and because the homozygous red variety produces 100% or makes 100% functional protein, but this heterozygous produces only 50% of the functional protein, and so also it produces 50% the pigments, and the mixture of the pigments, white and red, resulted in pink color in the F2 generation. So this is a molecular explanation. So it is based on quantitative way, that is how much amount of that functional enzyme produced, accordingly how much amount of pigments produced. So 100% pigments are produced by the 2 copies of normal allele, that is R1, R2. So in the case of heterozygous condition, only 50% of the functional protein is produced, which produces only 50% of the pigments or half of the pigments of the parental type. So it becomes pink. So we can explain the coloration of the flower based on the quantitative method, based on the amount of pigments, based on the amount of proteins and enzymes produced. That's why I mentioned it is being explained based on quantitative way. What are the other examples for incomplete dominance? One in the case of plant by name Antirrhinum majors. It's normally called as a snapdragon or dogflower. Here also the flower color red and white, the parallel variety. When crossed, we get F1 pink as in the case of 4-o-clock plant, the same explanation. Now in the case of animals, in one fold variety, what is called Andalusian fawns. This is one type of fold variety where you have the feather color either black or white. When these 2 pure breeding parents were crossed, we got blue in the F1 generation and intermediate color. So that is otherwise called speckled type. So this is also another example for incomplete dominance. One in the case of animals, feather color. The F1 hybrid shows what is called the blue color and intermediate color. So in the case of complete dominance, any one of the parents appear in the F1 generation. In the case of incomplete dominance, intermediate character appears in between the 2 parents. So the character appear is in between the 2 parents. In complete dominance, the character appear is resembling any one of the parents. We have to go for codominance next type. We'll talk